So, okay. So liverworts, here you see there's a family tree of bryophytes on the left. The ones in the green boxes are all the groups of liverworts. They're extremely diverse from the point of view of their morphology. The structure of the gametophyte is much more diverse than in mosses. You have leafy, you have thin ribbon-like thalli, you have thicker ribbon-like thalli, and then you have very thick ribbon-like thalli, and then some that are really sort of weirdly in between. But what unites them all, there are a couple of structures. All liverworts have structures in their cells or their membrane-bound organelles called oil bodies that contain terpenoids of various sort, often unique compounds. Um, and those might be inclusions in a regular cell that has chloroplasts and all the other organs, or they might be in a cell that's empty of everything but just a great big oil body. So all liverworts have those. They also all have a similar mechanism for opening up their capsule. Now at the top, you see there's a moss capsule, and you know it has an operculum and a peristome underneath that opens and closes. In liverworts, we just have a simple valve-like four valves that split open when the capsule's ready to discharge. It puts forth this mass that looks like cotton candy, sort of, which is a mixture. Looking microscopically on the right hand, there are spores, and then these long structures that with these coils of thickenings called eliters, and those act like springs, and they kind of propel the spores out into the environment. So as different as they all look, all liverworts have those things. We're going to focus on the two most common groups tonight. I'll have one example of some dissection of each. Marcantioid. Marcantia is the liverwort that everybody knows, where you have a thick thallus that's got a number of layers. Here's a cross section. There's an epidermal layer, some chlorophyllose layers, which are stained purple here, and section of thin celled walls full of water that are sort of like the parenchyma of a leaf, and then an underside. And the sexual organs are borne on stalks under the sort of the arms of these sort of umbrella-like structures or on top of them. The leafy liverworts are much more diverse. We were talking about this a little earlier, where you have a stem and you have leaves along it in two or three ranks. Two of those ranks of leaves will be identical, and they'll be not exactly parallel to one another. They'll be offset. They do go around the stem in a pattern, but the placement, they're all lined up within each row. When you look down on the top of them, you see three rows only, or two, because this third row of leaves may be absent. Here's a stem where there is no third row of leaves, and here's one where there is a third row. Sometimes that third row of leaves looks identical to the lateral leaves. Sometimes it's very different, as in this case. But in all cases, in almost all cases, liverworts look flatter than mosses. This is a case here. Because the leaves are not arranged like a bottle brush, they're arranged in these two ranks primarily, with a third smaller rank perhaps. Huge variation in liverworts about the shape of those lateral leaves. They may be entire like these. They may have little lobes, or they may have lots and lots of lobes like this trichocolia, which kind of looks fuzzy when you see the plant. They also may have lobes that are folded and pressed one lobe up against another. Here's a case in radula where we have bilobed leaves where the smaller lobe is pressed to the larger one. And then in Frulania, we saw Frulania earlier in Rice's presentation, the little lobe has through evolution been turned into kind of an upside down flask. There's all sorts of fantastical variations that could be made from those flasks. Anyway, I'm going to show you preparation of two with a leafy liverwort called Scapania, and then a thallose liverwort called Marcantia quadrata. Before I do that, I'm just going to show you some of the tools that I prefer to use. Everybody's different and will pick out their own, you'll find your own way to the tools that you like. I like a pair of number 5 Dumo Star, very sharp, very fine pointed forceps. They break very easily. I can't tell you how much money I've spent on these over the years. They're $35 a piece on Amazon, and I always have several in reserve because all it takes is one drop on the ground or one poke into the side of your microscope and it's ruined. I also use a double-bladed razor blade because I like the flexibility of it, but yes, I have shaved off many layers of skin off my fingers while doing that. When I put them on, I use obviously a microscope slide and cover slips like everybody. I have a supply of what are called depression slides. I inherited these in grad school and I've had the same 10 or 12 ever since. They're slides with a depression in them. And I use those to for my initial sorting and wetting up of things. Like I say, not everybody uses the same tools. My husband and I share a home lab, and I just took a picture of some of his tools. You can see some are the same, but he likes curved forceps. He works on fungi, so we don't work on bryophytes, but the tools are the same. He likes curved forceps and a needle, and he uses a single blade, razor blade. He uses a watch glass instead of a depression slide for initial wetting. And that's this bottle of immersion oil, I think is probably 40 years old and it's still half full. So you could buy one of those for looking at things under 100% on 100x. It'll last forever. I'm very picky about my water bottles because I want just a very small drop of water. I can add to it, but one of the most annoying things is to put too much water on a slide and then you put your leaves or your sections and either there's so much water that you can't focus well on them or the things that you most want to see go floating out from underneath the cover slip. So these I bought fairly recently on Temu, $3 a piece, but I had to buy six and they water bottles with a very fine point. The pictures I'm going to show you, the videos and the slides were captured with, this is my new toy. It's an Elecliv digital microscope. It was my Christmas present to myself, cost me a little under $200. And I'm getting to love it. It's still a little awkward for me, but it works well. It takes both stills and video. It's pretty easy to use. Then, of course, my compound scope. I have an AM scope with a little camera on it that attaches. I can move the camera from my compound to my dissecting scope, and then see the pictures on my computer. Our first candidate is Scapania subalpina, which can be very common along little streams in the mountains, the Front Range of Colorado. This is along Geneva Creek near Guanella Pass. That's what it looks like a little closer up. And here it's close enough up that you can see that there's a stem and we have bifid leaves that here's one of those cases where if part of the leaf is folded against the other part. So one of those, in this case, the dorsal lobe is smaller. Now, this is just showing this may be too elementary. I apologize if it's too simplistic, but starting with a dried collection, I don't like to wet up the whole collection. I like to look for a section of my specimen that has good typical stems and then pinpoint a few stems because I'm pretty convinced over time that when you wet up a whole lot of material, each time you do that, it doesn't quite come back. It becomes a little bit more mushy and hard to work with. So I pick out what I want and I'll pick out 5 or 6 stems. Let's see. Why don't I go on to the next one now? And then I'll collect some stems in one of the wells of my depression slide. And of course, I've managed to bring a whole lot of dirt over. And so the first wash in the depression slide is to get rid of a lot of that grit. And then I'll go ahead and I usually have more stems than this that are candidates for various things that I want to do. Use some to look at leaves, some to maybe make a cross section of a stem, others to maybe mount whole, if that seems like the thing to do, depending on what it is. And then I've moved them here to the other well, and then I'll do some more cleanup, kind of swish them around, try to get rid of as much of that dirt. Most of that dirt is stuck in the rhizoids. And at some point I'll, start to pull it apart, trying not to pull the stem apart while I'm doing it. Now, there we go, pulling some off. Okay. Okay, so I'll decide those stems are for different purposes. One I might make, if in this case, this particular genus, stem cross sections doesn't really help. But what is important is to see this leaf, these folded leaves, to see them flattened out. And because of the way they are, I really have to look, oops, I have to look at it with a through the compound scope, and I have to remove the leaves in order to do that. So I chose this particular stem for leaf removal. I'll grasp it with steps and try to pull off, I don't know, 5 to 10, depending, leaves that I will then mount on the compound scope. So there I'm pulling them off. And it's almost impossible to get in some, well, for me, it's often impossible to get absolutely the whole leaf. So I'm going to save a stem so that I can look at the leaf attachment on the stem, because once I've pulled them off, I'm going to miss some features. I'll say more about that in a minute. Here's what I mean. So in Scapania, it's very important to see whether or not the ventral the attachment of the ventral lobe is decurrent or not. And it's very hard to pull off the leaf in such a way, or at least it's hard for me, so that I would get that decurrency. But by looking through the dissecting scope and going up as high as I can in magnification, I can see here, you have to maybe take this on faith, but there's a little line that runs down, and that's the decurrent attachment of the leaf. And it's not as dramatic as in some species. In some species, it might go all the way down to the next leaf, but it's truly, there is a part that's going down here. And so I can say, yes, indeed, that is decurrent. So I took that leaf, the leaves that I pulled off, I mounted them up, and from this large, the whole leaf, I can take some critical measurements, compare the size and the shape of the lobes, which is critical. I can also look at the apex and the margin, see the marginal cells, how big they are, whether or not they're teeth on the margin, whether or not they're gemmy at the apex. And then even higher magnification, I can look at oil bodies. That's what these gray, sort of grayish spheres are. And I actually cheated and brought in a different species here, because another thing that doesn't show up so well in that slide is the thickness of the walls between the cells. These corner regions are extremely important in liverworts. The species level usually, whether these corners, they're called trigones, whether they're small or large, or whether they're bulging or not. Okay, moving on to Marcantia, or as it's been known, Pricea quadrata. It's now called Marcantia, and it's typical. You've got these stalks with the female reproductive structures and stalks with the males, and then the surface of the thallus is sort of like reptilian kind of. And this is what we're going to focus on. They're so pretty when they're alive. When they're dead, not so much. I think maybe this is why I don't really like working on these nearly as well. But again, I've put a drop of water on, and here's, so you can see the sporophyte is laying across. So here's the thallus is in this area. And I'm going to reach in and pull off some pieces. I kind of mangled that one, but I will eventually get it here. Key being, I'm going to need to make sections of part of that thallus, but I don't need the whole thing intact for that. Here's a couple of pieces that I pulled off. You see you've got the upper surface here. It has purple scales on the underside. I'll say more about that in a minute, but that's what you're seeing there. Okay, and here's my piece that I'm going to section. And so I'm going to grab it, and I say I'm still learning how to dissect under this this new toy that I have, and it's, well, sort of excruciating to watch. The point being, though, that if you're new to it can be so frustrating, but you have to just keep at it. Your first couple sections may be terrible, but if you just relax and keep doing it, you will probably eventually get something that you can use, even though maybe your first ones are really thick. And it seems, but I've also, there's a lesson there that even thick sections and sections that aren't very good might still tell you something. So you just keep at it, and I'm not going to torture you anymore with that. But eventually I did get a bunch. some that could be used. And I spent a little time separating them out. I think it's not that it's that critical, but I tried to get them all flat and then also in the middle of the slide in a single drop of water. And if the water isn't enough, I can always add in a little bit from the side. But again, I don't want my best section to go floating off the slide or off out from underneath the cover slip. Let me see. Now here's an example. It's a terrible section. It's very oblique, but it just happens to show a key feature for identification, which is the nature of these pores. These are the pores in the upper side of the thallus, up surface of the thallus that gave it that kind of odd scaly appearance. And I can see right away that they're formed of concentric rings of cells with an open space below. Well, that's a great clue. Even though it's a terrible section, it's a great clue. This is the upper surface, here's the lower section, but it tells me some stuff I need to know. And when I zoom in on that and look down, I can see that there's actually, it looks like there's actually 5 cells in this ring, that this is the pore opening. And here's a better case where I got a section right through one. You can see here's one of these pores. There's a row of cells. They describe them as being barrel-shaped. And then down below, there's a chamber, kind of an open chamber, and growing out of those chambers are filaments. And that's enough right there to get me pretty far along in the identification. Okay, that's all. I'm going to stop right there. And if you have any questions, happy to answer.